So, good afternoon, all students and colleagues. So, we'll be talking and revising a bit of clinical research, and of course, randomized controlled trial is our main topic. And before we reach the topic, we need to understand the nuances of how the research has developed and why we require RCTs to part of introduction. The clinical research, as you all know, is a branch of medical science. Clinical trials are the experiments, they are conducted on humans, and these are experiments. See, both are employed to determine the safety and effectiveness of medications, devices, diagnostic products, nutritional behavioral changes, and treatments that are intended for human use. And randomized controlled trials are used to establish the safety of many interventions. So, if you know that clinical research is a structured process of investigating facts and theories, it's a systematic way to examine a clinical condition and outcome and intervention, and it helps in generating evidence for decision making to improve methods of delivery of health care. So, RCT is a very important aspect of this. A clinical trial is also known as a patient-oriented research, and so the outcomes are usually patient-oriented outcomes that are important, not the surrogate outcomes. Health-related interventions are evaluated in clinical research in respect of drugs, radiopharmaceuticals, procedures, devices, genetic therapies, natural health products, preventive care, manual therapies, psychotherapies, and drug metabolism, etc. So, all these, there are various types and forms of clinical trials with methodological differences, but the ethical principles and procedures are the same and are applicable to all. And these are very important to understand that the history of clinical research has to be kept in mind for all the researchers, and the important aspects I'll briefly touch. It's got a long and rich history, and Ayurveda dates back to 1000 BC and is the foundation for modern science. Even the traditional Tibetan Chinese medications have roots in Ayurveda. You'll be surprised to know that Charaka Samhita is a treatise on general medicine, and Charaka knew about 500, and Shishrada know about 760 medicinal plants. Ayurveda offered recommendations on diet, herbs, music, massage treatment, and meditation to restore body balance. It is a science of life encompassing body, mind, and spirit. So before we reach randomized controlled trials, you need to know the checks and the tragedies that happened with scientific experiments. The first reported experiments started 600 BC under the king Nebuchadnezzar II, when the children of royal blood and children of Israel were given diet rich in meat and wine and diet rich in legumes and water respectively. And after 10 days observed improvement in wisdom and understanding, a study was carried on for 3 years, and the superiority of diet rich in legumes and water was established. This was in Old Testament 600 BC. That's how the clinical research first reported. And Ambrose Paré, the French surgeon, studied the boiled elderberry oil for treatment of gunshot wounds. When he ran out of this oil, he made a mixture of egg yolk, serpentine oil, and rose oil, and this mixture yielded a better relief. That's another evidence that is generated in 1537. That's how the research has progressed. And the famous of all is James Lind's experiment, systematic experiment in a modern era on the decks of the ship Salisbury, when the seamen developed scurvy. What he did was he chose 12 seamen, divided them into 6 groups of 2 each, and treated with cedar, vitriol, that is sulfuric acid, vinegar, seawater, oranges and lemon fruits, and barley water and nutmegs. And, so that has proven that scurvy can be prevented by vitamin C oranges. Austin Flint was the first used placebo in the treatment of rheumatic fever in 1863, and the idea of randomization surfaced in 1923. New system of drug regulation, the safety of new drugs started appearing in 1938. the Nuremberg Code following the Second World War, the Declaration of Helsinki in 64, and the Food and Drug Administration, USA, the International Conference on Harmonization, and the Common Technical Documents of 2000. All these are checks. A lot of refinement of research methodology and clinical practice happened in the last 30 years and closely govern the outcomes of randomized controlled trials. So the history of ethics and disasters also you should know a bit before we touch on RCT. The first one is the a significant part of the ethical regulations in clinical research is necessitated by these disasters. So all these are important to understand randomized controlled trials where informed consent is important and ethical committee clearance is important. In 1937, sulfonamide cold syrup elixir tragedy due to ethylene glycol preservative surfaced when children died, and that led FDA in 1938 to promulgate an act which drafted to create new guidelines for drug control and safety. This act not only defined the conditions required for release of new medications, but also served as a stimulus for medical research. Any drug which you are using has to be tested in randomized controlled trials to achieve a to prove its efficacy and later on its effectiveness. FDC Act 1938, that is the Federal Food, Drugs and Cosmetic Act, US Disaster Centers. So that is a response to that disaster. In 1915-60s, we all know that there's a drug called thalidomide, which is as popular as aspirin to treatment of almost every health condition. It also was used to treat pregnant women for their morning sickness, and ultimately they found out that phocomelia resulted in the use of this drug, and that has this thalidomide tragedy, unethical courts necessary for physicians, also for production of participants in clinical trials worldwide. So the Declaration of Helsinki 1964, which was reviewed in 75, and that led to review of research proposals by independent review boards. All the RCTs are to be subjected to review by independent review board. When we know what is RCT, then you'll understand the significance of this. One more story I will tell you, that is deplorable Tuskegee study of untreated syphilis cases in Negro males. Alabama, US 1932-72. The sufferers were denied the standard of care of injection penicillin available at the time to study the natural course of the disease. So Belmont report in 1979 appeared, and with that the Institutional Review Board informed consent and respect for person. There should be no deception or misinformation. Beneficence, do no harm, justice. All these things were incorporated in the Belmont report, and which are very important for conduct of randomized controlled trial. And the German race used for treated as guinea pigs that led to Nuremberg Code. Following this tragedy, principles of ethical research were established under the Nuremberg Code. It outlined 10 critical aspects for the conduction of experimentation with humans. So that based on the lessons learned from the major disasters in creation and commercialization of novel therapeutic drugs, the process for the development of a new drug or a device has been standardized, and safety has become a major issue. Though experimental studies go through the following stages: preclinical phase 1, 2, 3, and 4. And we'll just touch briefly on the anatomy of methodology of clinical research in 2 or 3 slides. The clinical research can be viewed at two different viewpoints for learning and understanding, but these two themes run together. Anatomy of research deals with the research question, design, subjects, measurements, sample size calculation. They all should be summarized in protocol. The researcher's goal is to create these elements in a form that will make the project feasible, efficient, and cost effective. The second theme is the physiology of the research, how it works. Study should yield valid inferences. First, what happened in the study? Secondly, whether the findings can be generalized to the population outside the study. So the goal is to minimize the errors. There are two errors. There is random error and systematic error, which can threaten the conclusions based on the inferences derived from the study. So the design should solve some of the issues, and randomization is a very important aspect in this context. So the anatomy of research question, background and significance, the design of cohort cross-sectional case control has already been dealt, and RCTs will deal in a couple of slides later. Study subjects, variables, statistical issues, all these come under protocol. The physiology of research, whether research is conducted properly, giving it internal validity and external validity, implementing the study, the causal influence, associations between a predictor and outcome variable in order to draw a causal influence, whether the research has taken care of the errors in the research, that is random error and systematic error. Now comes the randomized control trial. The structure of the clinical research you understood as the research question, design, subjects, measurement, sample size, and how it works is what happened in the study, whether the conclusions based on the study results are free of errors, the random error and systematic errors which threaten the conclusions, and based on these, the internal validity is judged. Can the results be generalized to people outside the study? So these are some of the issues which are important in our understanding. So study designs fall as for all of you into three main categories: observational descriptive studies, observational analytical studies, experimental studies. Here the researcher intervenes in some way with one group of people and reports any differences in the outcome between the experimental group and a control group. That means there are two groups. So these studies are experimental studies, are uncontrolled trials, controlled trials, randomized controlled trial, crossover trial, N of one trial, and factorial study. So the types of study, as you know, what is happening in the study? Are we looking at a group? Is there an intervention to some or all the subjects in the group? If there is there a control group? If is there a random selection? If there is a control group, or if there is no control group, the inferences are like this. If there is no control group, it is called observational study. If there is a control group, it is called observational analytical study. And observational analytical study are further divided into cohort studies, case control studies, and cross-sectional studies, which have been dealt in the previous classes. The randomized control trial is if there is a random selection, there is a randomized control trial. If there is no control group, there is a observational study. And these are the types of studies that we come across in our research. Okay, so these things are important for one thing, that is fair allocation of healthcare. Let us take an example of von Helmont in 1662, when bloodletting was the treatment of choice. Then what he said was, he's opposed to this treatment, this particular mode of intervention. He said, let us do a study. We divide the patients into two halves, and let us cast lots that one half of them may fall into my share and others to yours. You do bloodletting, I will do some other sensible thing, and we will see how many will go to funeral in the end. So that is a 1662 thought process. So the experimental studies of Lind, I have already told you that what he did, and how the oranges have come into the vogue. And so what is random? Random is, in a layman's term, having no specific pattern, lacking causal relationship, and haphazard, random acts of nature. But scientifically, what is random is a process in which there is associated with every legitimate outcome a probability. So probability is associated with random, a process in which there is a probability of having heads or tails in a coin toss, in a dice throw, in a drop, in poker, etc. So these are very important. And Donald Fisher in 1890 and 62 used this first method to allot plots in a agricultural land, to make it sure that there will be no errors in allotment. So so that everybody has equal share of all the problems of the plots. And he used the randomization technique, that is Ronald Fisher in 62, and Bradford Hill, his epidemiologist, who has said that in aspects, the reactions of human beings are under any circumstances extremely variable, and that is where the trouble begins. And having used a random allocation, the sternest critic is unable to say that quite probably the groups were differently biased through our predictions or stupidities. So random allocation is a very important process in generating. The first trial of randomized control trial was for tuberculosis in 1946, and streptomycin is on premium, was not able. So it should be fairly distributed, and the risks and benefits to be assessed and fairly allocated for limited resource. So they did a randomized control trial, and that's how the first randomized control trial was done, and streptomycin has been proven to be very effective. So are RCT still the gold standards? Yes, we'll see what is an RCT. It is a framework for evaluating evidence. Compare the results of our randomized control trials with non-randomized studies. There are in literature many such examples. One is vitamin supplements. High-profile cases with conflicts between trial and observational study results are vitamin supplements are useful in preventing the cardiovascular disease, which has been proven a myth. Women's Health Initiative regarding hormone replacement therapy and ischemic heart disease, which was also an observational study which has shown positivity, but which has fallen flat after randomized control trial. So there are errors in research, there are confounders, there are biases. So we should know before RCT what are the results, the bias and confounding factors are there. The results will be wrong. If they are avoided, the results will be correct. So no study, understand this, no study is free of errors. The erroneous research inferences are mainly due to random error, which can be corrected only by randomization, systematic error, which can be corrected only by a proper research technique. The erroneous inferences that can be addressed and prevented at three stages: at design stage, at the implementation stage, at analysis phase. Also, these inferences can be addressed and prevented. So if you design a study and implement a research question, and you make a study plan, and if it is a randomized control trial, it can externally validate that, means externally can apply generalized categorization can be done. And if your study plan is implemented with no mistakes, the internal validity is improved and actual study is done, the findings in the study will be without errors, minimal errors. The inferences are close to the truth in the study, and then that truth in the study will be close to the truth in the universe, and we can draw conclusions. So there are two terms here: external validity, internal validity. Whatever study is done with proper research technique gives the study internal validity, and external validity is if it is a randomized control trial, the results can be applied to the population from which the study is done. So external validity is methodology of a study. So mistakes, they do not happen by chance. So it must be reduced by a good research technique. This is important, as once a mistake has been made, it cannot be measured or controlled by using statistical techniques. The systematic error causes can be design flaws, protocol flaws, subject limitations, mistakes in data entry and analysis. So observer, instrument, and subject errors can creep into this research. Now, what is the other problem? There's confounder, where a relationship between two variables is attributable to a third one relating to both, but not on the causal pathway. It makes it appear as if there is a direct relationship between the exposure and outcome. Association that would otherwise have been present. The problem arises when confounding factors are not identified by the researchers. Randomization only will prevent. So exposure and outcome, the confounder is standing here. Drinking coffee can cause lung cancer. Smoking and can cause lung cancer. And people who drink coffee smoke a lot. So this is a type of analogy that is given for confounders. There are positive confounders in which the dissociation between two variables are not associated. They overestimate an association. The negative confounders underestimate an association. So these are to be though. So the confounding factor bias you should understand before randomized control trial. The confounding factors differ from bias. Confounding is not created by some mistake made by the researchers, but it cannot be controlled by better research techniques. It can only be controlled by, must be identified, eliminate, and these confounders should be spread equally between different arms of the study or neutralize their effects on the results using a statistical techniques. Confounders can be measured and controlled for, but good study design is an essential as one cannot exclude a confounder which was not identified and measured. So dealing with confounding, randomization, restriction, matching at the design stage, restriction, stratification, multivariate regression at analysis stage is useful. That's how you deal with confounders. And the errors in research, the probability of getting a false positive, so the null hypothesis, rejecting null hypothesis when it is there is a type 1 error, it's called alpha. The level of statistical significance in calculation is 0.05. That is 5% maximum chance of incorrectly rejecting the null hypothesis. The probability of making a type 2 error, failing to reject the null hypothesis when it is false, is called beta. And 1 minus beta is the power. Power is the probability of rejecting the null hypothesis in the sample. That means the experiment is true. The actual effect in the population equals the size. So if you want to see that, truth in the population, association between predictor and outcome variables, no association. Null hypothesis says that there is no difference. If it rejects, that is there is a possibility there can be a false positive error, and that should be minimum. And fails to reject null hypothesis, type 2 error is false negative, and uh, it has to be correct. So there are two true positives, true negatives, and type 1 error is false positive, type 2 error is false negative in any studies. Type 1 and type 2 errors are unavoidable, that can be minimized. So ideally alpha and beta should be 0, but it is not possible to avoid them. The maximum chance of making type 1 and type errors is established in advance of the study or a priori. Alpha to be kept as 0.05 to 0.01 to 0.10, and beta is 0.20, a power of 0.80. So it can vary between 0.05 to 0.20. So low alpha, it is important to avoid type 1 false positive error, and low beta small effect size, it's important to avoid a false negative error. So depending on the situation, we want this false positive and false negative errors. And so observational studies have many predictor variables: age, sex, race, smoking history, etc. Many outcome variables like heart attack, quality of life, unpleasant odor, etc. But clinical trials are different. Predictor variables is an intervention is manipulated, and the effect on the outcome variable is observed using randomization to control for the influence of the confounding variables. So you will always have two groups. So the observational studies, there is no randomization, no masking, and generalizable to maybe representative group, more heterogeneous, real less expensive, unintended outcomes. Whereas randomized trials have what if there, but the disadvantages of the observational studies, which can be overcome by randomized controlled trials, are selection bias, unknown confounders, residual confounding, cohorts are select two, systematic differences due to treatment due to bias, information bias. So unmasked assessments, follow-up protocols, expensive survival bias. These are the things. So what is the rationale for randomization? It avoids selection bias. Prognostic factors related to treatment assignment, confounding by indication, that is called is avoided, can influence outcomes as strongly as or more strongly than treatments. Tends to produce comparable treatment groups, known and unknown confounders, assure statistical test, t-test, chi-square test. They have valid significance. And defined time point for trial entry. These are randomization. So there is a lot of internal validity. It avoids selection bias. It avoids confounding. It controls similar to the experimental group, and there will be no difference. Reduces information bias of masking protocol, detects small to moderate effects, eliminates bias, that is timing of treatment. So the opposite is what is the problem? External validity is always a select population. This trial is done. If it is a multi-centric, multinational and trials, then it can be generalized. Otherwise, it's difficult to generalize. Can't evaluate potentially harmful things. You can't deny treatments, as we've seen in some of these, and equally all to be treated. If there is a gold standard treatment, we cannot conduct RCTs, often too small to detect important side effects, too short to evaluate longer term effects, and expensive because RCTs cannot be done for longer time than few months. So randomization, if researchers and our subject decide allocation groups, bias may be introduced. So different groups may differ in a systematic way so that the results across the groups cannot be compared. So allocation should not be with the researchers and our subjects. There may be differences in the subjects who experimental treatment group and those who don't want to be are not selected to be in the experimental treatment group. So no randomization introduces a bias. So randomization ensures that all the entering a study have an equal chance of being allocated to any group within the study. The allocation cannot be predicted in advance. It minimizes some types of bias also. Random sampling also is the basis of statistical theory. If there is no randomization, the results may be misleading. That's the importance of randomization. The method of randomization can introduce bias also. It is therefore important to know how the randomization was carried out to be able to assess whether this is likely to influence the results. The generation of a random list can be helped by computer random number generator, random number tables, shuffled cards or envelopes, etc. Methods of randomization. Simple randomization, each subject's treatment at random as they enter the study independently of any other factors. Generate a list of random numbers between 1 and 100, 53, 76, 34, 88, 12, 59, 96. Decide the cutoff for treatment or control group. More than 50 will go into the treatment, less than 50 will go into the control group. So you can see that more than 50s, that is 53, 76 are going to treatment, 34 going to control, 88 to treatment, 12 going to control, like that. This is one strategy of simple randomization. And so then the treatment group and control group, you can subject wise, you can do other methods of randomization, right? So methods of randomization is an important these strategies of block randomization. Subjects are put into blocks. Subjects within each block are split equally between the treatment and control groups. Decide on the size of each block. For randomization, there are methods. Randomization, shadow block randomization, where you can select sequence 1, sequence 2, sequence 3, sequence 4, and when the subject 1 comes in the first block, it goes to treatment, subject 2 goes to treatment, subject 3 goes to control, and subject 4. If second block, the thing order has been changed, the subjects as entering in this second block will have a different allocation. Similarly, third block, like that, the block randomization uh, procedure is gone. And block randomization examples which you have seen now, and I have explained to you. So the treatment and control groups, equal numbers will occur in block randomization. Dealing with confounding factors, stratified randomization, you require male and female separately treatment groups and control groups. So you add another gender factor also into that. So subgroups are formed in stratified randomization. Within each subgroup, block randomization takes place, and the confounding variable is equally distributed in the treatment and control groups. That is the of stratified randomization. So men are placed in the subgroup for men and women are placed in women subgroup. Then randomization takes place in each subgroup as shown here. And this how the stratified randomization technique takes place. And if a sex is a confounding factor, and both sexes are equally distributed after stratified randomization. There's one more aspect of this thing, that is allocation, concealed allocation. Randomization allocates subjects into groups. The researchers want the best subjects in the new treatment group so that the results are favorable. If randomization schedule is published in advance, researchers may be able to put their subjects into the new treatment arm, which is their favorite. So concealed allocation protects the randomization. The treatments given in the kept as secret. The subjects, researchers identify the treatment arm and the control arm. Both the subjects and researchers, they don't identify. A vital part of the randomization process is this. How do you do it? Centrally concealed, telephone, pharmacy concealed. We don't know what is there in A, packet B. And centrally, if they say you allot to number one treatment, number two treatment, we don't know what is there in the treatment. Sequentially numbered, opaque, sealed envelopes can be used. Numbered coded bottles or containers can be used. So concealed allocation will be done with your own methods and how you can do. And so that's the pre-process. Now let us study. Then you have population, you identify the target population, you identify the eligible population, then you have study subjects. So when you do study on the study subjects, it will be extrapolated into the target population. So the investigators select a sample from the population. He measures the baseline variables, randomizes the participants, randomly assigning the intervention, can eliminate the influence of confounding variables, applies interventions. One should be blinded if possible, measures outcome variable during the follow-up of blinded to randomized group assignments. The chap who is doing the outcome assessment is blinded to the assignment. So you can see the population, the sample, the treatment is given, the placebo is given, and the present and the future. This is present, this is absent, this is present, this is absent, and you have to do a contingency table analysis for this. So this is the basic structure of randomized controlled trial. So populations, target population, you have excluded with somebody with exclusion criteria, you have included and brought in the eligible patients, then you selected a study participants depending on sample size, and you have generated the results which are extrapolated onto the target population. That the randomized controlled trial is done. So research question, target population, the design, random and systematic errors taken place in the intended sample, and you have implemented the study. Again, there is a chance for random and systematic error, which we have to minimize. The actual subjects are selected, actual measure intervention done, actual measurements were done in the study, and the intended variables are studied, and phenomenon of interest as a truth in the universe has been identified. But the problem is we have to minimize at both design and implement stage the errors which can interfere with inferences. So let us see how randomized control trial looks like. We've done a target and restriction and so the randomization process is preserved. And after this, what you do is concealed allocation. After randomization, concealed allocation. Okay, before that, you restrict your match so that some biases are avoided. Then you want to take care of a trial region. Blinding of doctors and patients takes place. Blind of doctors and patients. The trial ends. Blind assessments of outcome. Now we have to dropouts. You have to calculate, and of course you have to on both arms you have to calculate the dropouts. And the placebo arm is unfolded now because blinding has been unfolded here. And after that, after the allocation is revealed, the intervention arm results and the placebo arm results are there. So what you see, the blinding and masking is an important aspect. Single blind, double blind, triple blind, quadruple blind can happen. Blinding using the terms doesn't guarantee correct implementation, but describe who is kept unaware of what. Describe when decoding is done. Sometimes it's not possible to blind, then accept it. Endpoint, measurable endpoint should be selected in study, and clinical endpoints like mortality, disability, disease progression, improvement of symptoms in patients, quality of life measures. As I said, clinical trials are patient oriented. So you should select clinical endpoints only: mortality, disability, progression of disease, etc. The surrogate endpoints are predictive of important clinical outcomes. They are predictive of outcomes, but the relationship is not guaranteed. Whether there is a drop in blood pressure that is going to save the individual, we are not sure. So you should always concentrate on clinical endpoints, and composite endpoints sometimes may be taken care of. So in essence, when we are doing assessed for eligibility, this is a consult statement. Some are excluded, not meeting the inclusion criteria, declined to participate, other reasons. You enter the number here. Now, how many are assessed for eligibility? You number there. How many are randomized? You have to enter the number. Allocated to intervention, how many? Received allocated intervention, how many? Did not receive, how many? Allocated to intervention, second one. Number and received intervention, did not receive intervention. Lost to follow up, discontinued intervention, you follow up, whatever happened, you have to mention in the consult statement. And analyze, excluded from the analysis, give reasons. And in both arms, you have to give reasons why they are excluded from the arms, and it could lead to the intention to treat analysis now. So we have a research question, we have a primary hypothesis, we have made two groups, and experiment started, and results are generated, and results are compared, and there will be differences seen in the results at one group, and there may be no difference seen in the result. This is null hypothesis. And null hypothesis is accepted if there is no difference seen in the results. Null hypothesis differences are due to chance. So whatever differences that are seen can be because of chance. So if null hypothesis is accepted, consider type 2, right? If consider differences are due to chance, consider type 1 error. P is calculated, significant testing has been done in the statistical arena, and if it is more than 0.05, what do you see? Differences are due to chance. If it is less than 0.05, differences are not due to chance. So that's the basic. Okay, preclinical development. If any drug or vaccine is there, biological target is identified on 19 SARS-CoV-2 infection, new drugs come, it tested in the laboratory models, and this is called preclinical development. Then human testing, phase 1, when and phase 4 is effectiveness after the controller gives approval. So phase 1, intervention in humans, first stage in testing a new, usually 10 to 30 people, identify tolerable dose, provide information on the drug metabolism, excretion, and toxicity. It is, if toxic is often not recommended. Phase 2, usually 30 to 100 people, preliminary information on efficacy, additional information on safety and side effects. Usually 100 plus people in phase 3, all assesses efficacy and safety. It's a controlled trial, usually randomized controlled trial, and that's how the phase 3. So, and parallel, there are various types: parallel, crossover, group allocation, factorial, large sample, equivalency, non-inferiority, adaptive. So various trial designs are there, which are not going to go into the details. Equivalence design means here intervention response falls sufficiently close to the control group response. How much difference can be allowed between the treatments to be considered equivalent? Detectable difference should be small. Probability of detecting a difference should be large. Therefore, a large sample size is required here. Sometimes it may be infinity. Flipped as compared to superiority, there is a difference between the two treatments, that is the null hypothesis. Alternately, there is no difference between the treatments. So type 1 error showing no difference when there is one, type 2 error showing a difference when there isn't one. So this is how the hypothesis is understood. And non-inferiority design is determine whether treatment A, frequently a new treatment, is at least as good as treatment B. Test if hypothesis A is worse than B, then one can be rejected, that one-sided. So operationally, the intervention response, if worse, is still sufficiently close to the established treatment response. So that detectable difference is small, but hypothesis is one-sided, so it doesn't require a large sample size as equivalency trial. That's why it is called inferiority trials are favored, non-inferiority design, as we can see in the example which I am going to quote in the relay trial. Prevention of venous thromboembolism after total knee replacement. They have studied apixaban and enoxaparin. Treatment continued for 10 to 14 days after surgery. Follow-up visits at 30 and 60 days after the last treatment. Main outcome was composite of asymptomatic and symptomatic deep vein thrombosis, non-fatal pulmonary embolism, and all-cause death during the treatment. It is designed to detect a non-inferiority that is defined as 95% confidence interval. A limit of RR is 1.25. That means both the treatments are 1.25 is also the experimental drug to be accepted as equal. You can see this is the superiority, this is the border of the confidence interval. This is a 95% constant interval. Experiment is better, control is better. The non-inferiority means even if the response is control wise this much, this is accepted as good enough. That's why it is not superior, but it's also not inferior. That's the understanding. This is a shown, and non-inferiority is shown here because it is less than this borderline. Non-inferiority is shown here. Here it is crossing this border, so this is not shown. This is also not shown. Here both superiority and non-inferiority is shown. That's how the treatment difference. This is the borderline. This is a non-inferiority station 1.25 in this particular study. So dabigatran versus warfarin in patients with atrial fibrillation is one of the studies in NEJM. The study objectives and research question can be framed. Dabigatran is a new thrombin inhibitor in patients with atrial fibrillation. Is dabigatran 110 milligrams or 150 milligrams compared to adjusted dose warfarin non-inferior to prevention of stroke or systematic thromboembolism, and the others complied with the FDA recommended calculation for the acceptable margin of non-inferiority based upon 50% of the treatment effect of warfarin in 6 prior RCTs. So there is a previous study. Design is prospective, randomized, non-inferiority, active control, open label warfarin, national trial on sites, 44 countries, and average 10 subjects per site. Patients are into 3 treatment groups: warfarin, dabigatran 110, dabigatran 150, followed for 2 years for efficacy and safety. And you have to identify the PICO in this. That is the research question. Our patients are with chronic atrial fibrillation more than 75 years. If there is diabetes, hypertension, or ischemia more than 65 years, they are at risk for stroke defined as more than one of the following: previous stroke or TIA, ejection fraction less than 40%, NYHA is more than 2 for at least 6 months. That's the heart classification. The intervention is dabigatran for the duration of study of 2 years, 110 milligrams and 150 milligrams. Control is warfarin, 1, 3, or 5 milligrams with a target INR to reach 2 to 3. Dose adjustment by local investigator, unblinded. Outcome is schedule of visits, stroke and fatal stroke, explicitly defined, ischemic, hemorrhagic, or unspecified were all included. Systemic embolism explicitly defined and included in the outcome. It's an interactive design. So the outcome, primary outcome is stroke or systematic embolism, sudden onset of focal. This has been defined clearly. What is the definition of stroke. So everything has to be defined a priori, what you're going to see. The outcomes were defined explicitly a priori. That's most important thing. The study design has been done, and so mixed non-inferiority attribute population documented on EKG, how it was selected, what are the inclusion criteria, what are the exclusion criteria of this, and there is randomization, central randomization service, randomization process, not described, but in the trial, ideal would have been simple, unconstrained randomization, concealment of allocation through IVRS at coordinating center, active control group, open label, no blinding. Experimental groups are subjects are blinded as far as dose is concerned. Investigator blinding is not documented. Minimization of bias not documented in this trial. So that's how the study is done with patients with non-valvular fibrillation. This is for eligibility 20,240. This is a consort statement, and the treatment group dabigatran 150, warfarin. There are people who are included in the study, and the treatments were given. Intervention as documented was given, and then the results are obtained after randomized trial. The groups are equally matched in both sides, and stroke happened of dabigatran 134, and out of 5,942 didn't have, and total of this, and dabigatran 110 is this much, yes, and the risk in dabigatran is 0.02, that is 150, 110 is 0.03, and risk ratio is this much, risk difference is this much, and the what I want to show you is this, the NNT part of it. The NNT part, NNT is about, you can see the NNT, it's going fast, right? So that is 100, NNT is 100. Okay, next. Risk in dabigatran, risk in warfarin, risk ratio, risk difference, and risk. Okay. So lastly, I would like to say that whatever is a topmost value, single randomized trial comes below that. Systematic review of observational studies addressing patient-oriented outcomes comes next. And at the bottom is unsystematic clinical observations, physiological studies. That's how the evidence that is generated is given. And this is a grade system of classification, which is to be used for generation guidelines, where the quality of evidence is decided. In randomized trial, the quality of evidence is high if the risk of bias is lower, and if a larger effect size is there, and a moderate quality of evidence. So all the trials in a in a guideline development should have the quality of evidence. That's what we are trying to say. The randomized controlled trial is the highest quality of evidence if it is done properly. I think with these few things which I think I finished the clinical research and randomized control trial. And thank you all for your patient listening. And I would like to have any questions if you have, I'd like to answer them. Are you all there?
Yes, sir.
So I think there are not many questions, not even a single one. Are you all there, students? Okay, then have a good day.
Yes.
I'll close the meeting.
Thank you, sir. Thank you, sir.
Okay, then great. Just